Hi, everyone. I'm Ginger Johnson, CEO of Define Health, and we welcome you to today's Define Health Insight Series webinar, Advanced Therapies in the CNS, Time to Get Smart. Define Health is a Cello Health business, and Cello Health is comprised of various subsidiary companies that offer advisory services across the development spectrum. Define Health itself is focused on value creation for early-stage biotech. Our strategic analyses bring a sophisticated and current understanding of the science, which is critical to understanding the potential value of a drug or platform and its competitive differentiation, thus enabling clear communication of that value to investors, payers, and potential partners. Today's webinar will feature Joel Sandler, CNS practice lead at Define Health, Mike Rice, practice head for Advanced Therapeutics at Define Health, as well as Stephen Gray, PhD, Associate Professor, Department of Pediatrics, University of Texas Southwestern Medical Center. You can review each of their bios in the event invitation. Before we get started, I'd like to bring your attention to the following events Define Health will be participating in this fall. On November 1st, Ed Saltzman will be participating in a panel at Mass Bio, organized by Cello Health, entitled Accelerated Early Drug Development, Compressing the Timeline from Clinical to Commercial. On November 28th, James Lee, co-lead of our oncology practice and heading up our BD effort in Asia, will be leading a webinar entitled Dynamics of Chinese Biotech Investments, Reshaping the Global Competitive Landscape. Finally, mark your calendars and sign up at an early bird discount for our 30th Annual Cancer Progress Conference in New York on May 7th and 8th. To learn more about these events and or about Define Health and our early strategic commercial development services, or about Cello Health more broadly, give us a call, send an email, or visit our website at definehealth.com. For today's event, please send your questions in as you think of them. We will try to save time at the end, but if we don't have time, we'll follow up with you directly in the next day or so. We anticipate that the slides and an audio-visual version of the webinar will be posted on the Define Health website within the next several days. Thank you, and please enjoy this edition of Define Health's Insight Series briefing, and I will now turn it over to Joel.
Thank you, Ginger. This is Joel Sandler. As Ginger mentioned, I lead our CNS practice here at Define Health. And I'm very excited to have everyone join us today for what promises to be an insightful and thought-provoking discussion. The topic, as Ginger mentioned, is Advanced Therapies in the CNS, Time to Get Smart. I'm joined today by Mike Rice, who is our practice head of Advanced Therapies, along with Dr. Stephen Gray, an associate professor at UT Southwestern. I'm going to lead off with about 5 to 10 minutes of intro slides, which will be available on the website, along with the audio of the entire webinar after today's discussion. And then I will launch into the discussion and start volleying some questions over to my esteemed panelists. So just to level set the advanced therapies trajectory, if one thinks about the traditional hype cycle, as many of us on the line are aware, it's been a wild ride and continues to be one, more or less following the classic hype cycle of initial exuberance, followed by or tempered by skepticism, most visibly with deaths from early generation therapeutic modalities in the space, as well as what in retrospect seems to have been a somewhat premium price for a product that was, I believe, the first gene therapy on the market in 2012. Since then, deal making has been percolating, and most recently with a very large deal in which Novartis acquired Avexis on the heels of what is some very striking data in SMA, which we will get into, as well as, previous to that, the first U.S. approval of a gene therapy product, Luxturna in Leber's, which we'll also be speaking to. But you can see at the end here this upward swing, and we'll get into, I think, a little bit later where that trajectory may be going in the future and some factors that could potentially have an influence on whether the field continues to thrive or perhaps is up for additional bumps along the way. So last week, Mike Rice, who I'm joined with today, along with our executive chairman, Ed Saltzman, gave an excellent presentation, which again is available on our website, and I highly recommend for anyone who hasn't heard it to go and listen, about gene therapy and advanced therapies, more from the biotech and pharma partnering perspective. And I really regard this discussion today as somewhat of a sequel, or at least an extension of that talk. And hopefully reasons for that will be more clear as we get into it. The initial, or one of the key takeaways that came out of that was positioning and potential for disruption of existing brands within either pharma or large pharma or specialty pharma. And 3 different therapy areas were identified as low-hanging fruit for disruption. I won't go through the details. Again, you can go back and listen, but suffice to say, these are franchises that were themselves ripe for disruption, largely because there had already been some branded franchises along the way to be displaced, potentially. What was not on that list are, broadly speaking, CNS disorders, though of course we can think of IEMs as influencing the CNS and ophthalmology as well. But broadly speaking, there aren't that many CNS brands or franchises to displace. And, you know, specifically genetic disorders of various sorts, proteinopathies, epilepsies, all of which could potentially be disrupted by gene therapy. But thinking about the trajectory of some of these other brands or franchises or therapy areas where there was, to some extent, incremental improvements going, let's just say, from A-track to cassette to CD and ultimately to digital. In a lot of these other CNS disorders we'll be talking about today, there aren't too many brands to speak of, and we really are going straight to digital potentially with these advanced therapies. It's not to say that the incentives are not lined up. They are, and really the stage is set for success in this space. And so I'll just speak very briefly to some of the different macro factors at play that that really do set this up for success. It always starts with the unmet need, whether we're talking about large heterogeneous disorders like Parkinson's, Alzheimer's, or rare diseases, which in the composite actually do comprise a pretty large or substantial group of disorders. Those rare disorders, as is illustrated on this slide, many of which do have a CNS component, whether it's neurons themselves within the CNS or secreted products or other pathways that have an impact, whether it's on the central or periphery nervous system. And in Defined Health's genetic disease database, for example, we've identified roughly 60% of disorders of one type or another that impact the CNS. There's plenty of white space, and so again, referencing that list of disorders, among those that do have a CNS component, when our analysts took a look at the pipeline databases, we're really only talking about a tiny sliver of those, 10% or so, for which there are therapeutic candidates in clinical development. So there's a lot of headroom, both within disorders that are being targeted today, as well as ones that have no candidates to speak of. The partnering activity, the M&A activity, as many of you are aware, is there. The investment dollars are there. specifically in neurology and specifically in advanced therapies that are focused on the CNS. And these are just a list of deals on the left that took place in 2018 alone. So we're talking about a lot of activity happening here, and more are coming in even today. The regulatory incentives are there, whether we're talking about the CNS specifically, or rare diseases, or RMAT designation, priority review vouchers, all of which really do kind of speak to additional incentives from the regulatory side to get into this space. So in the main, we're talking about a lot of different factors that collectively speak to potential broad value inflection across CNS targeted advanced therapies. So when we come back to this hype cycle and this question that I posed earlier about whether this upward trajectory will continue or perhaps not, one of the objectives of the discussion today that I hope you all come away with is that aside from clinical performance of things that are in the clinic today, a lot of this will be buttressed and ultimately, or even a priori, informed by sound strategic decision making, whether it involves positioning of programs within your pipeline, partnering and leveraging external innovation, smart commercial and decision making around pricing and market access that could ultimately benefit everyone in the end. And so really the discussion today is meant to be and meant to play off the idea that with all these different genetic disorders, many of which are monogenetic, although not all of them, that we'll be discussing today, there's an abundance of emerging commercial questions, but a lot of which needs to be informed by the technical side of things. And so here are a list of different questions that I'm not going to walk through, but I do hope that we can get into during today's discussion. And so underscoring that and the juxtaposition and the iteration between the technical side and the commercial side are our panelists today. So we have, as I mentioned, Professor Stephen Gray, who did his initial training in Jude Smolsky's lab and is currently research interest focused on AAV9 and other gene therapy vehicles and modalities as a means to address different CNS-related genetic disorders. And then on the commercial side is Mike Rice, as I mentioned, heads our advanced therapies group, and aside from his wealth of knowledge, also I believe serves as a surrogate for the many clients and different companies that he's speaking to very regularly in terms of understanding what the different commercial considerations might be. So I'm just going to then lead this up as questions to tee off the discussion, and maybe I'll start with a question for our technical expert, Steve Gray, who can start by introducing himself and maybe speak to what he sees as at a high level, the opportunities for gene therapy and advanced therapies, specifically in CNS disorders as discussed in the slides today. So Steve, can you start off with that?
Yeah, sure. So happy to be a part of this. As you said, my background is really centered around developing AAV vectors to target the CNS. I've done quite a lot of work with AAV9, published fairly extensively, and also involved in multiple IND-enabling studies and translation studies, moving treatments from bench to bedside. So that's kind of my perspective on this. It'll be a little bit AAV heavy because that's my background. But in terms of what opportunities are in the field, I think that you've seen really in the last 5 years a little bit of a revolution in the field of CNS treatments and specifically around CNS gene therapy. And a lot of that is owed to the emergence of a vector like AAV9 onto the field. And so there have been numerous preclinical studies that have supported the ability to deliver a gene broadly across the nervous system, either by a IV route or a intra-CSF route. And really just in the last couple of years, really exciting proof of concept that's happened with this vector in clinical trials. And I think that, it's easy to conceptualize where, if you see that you deliver a gene to treat a single disease and it's successful in humans, then overnight, I think it's opened up the possibility to treat hundreds of other CNS diseases using a very similar approach.
Great, thanks, Steve. And I think overnight is one of the key questions and topics of today's discussion. There really wasn't a market to speak of for this for a long time. There have been a lot of people, particularly on the academic and venture-backed early-stage biotech side, knocking away at this problem for decades, as I presented. But there are a lot of people rotating into this space, perhaps from elsewhere or orthogonal spaces, that are coming in, I presume, with questions and challenges in terms of understanding, what's the biology here? Is there kind of a one-size-fits-all, or is each disease going to be its own unique animal, to take one example? And then what's the business model here? So Mike, maybe I'll let you introduce yourself as well, very briefly, and speak to what you see as some of the high overarching issues of the day.
Yeah, sure. So I'm Mike Rice. Have about a 10-year background. If you go back to Joel's hype cycle, I have a 10-year background riding the wild sort of waves of the early stages of gene therapy development, going from initial proof of concept with Mike Blaze, French Anderson's, and Ken Culver's, clinical trials of ADA SCID back in 1989, showing, clear proof of evidence that gene therapy can lead to durable cures for, at least some primary immune disorders. And, since then, we've kind of rode the wave of a moratorium on clinical development on all gene therapy programs based on, futility in early trials and safety issues. And many of that, the technology was fostered by, principal investigators through the years, some large pharma companies like GSK that funded, advancement of viral vectors. Some of them have been long in the space and have decided that it might not be for them. Other ones have fought pretty vigorously recently to get into the space. What I would say is, driving, key change, and Joel had, touched on much of this in the introduction, is really that from a investment standpoint, a couple of years ago when we started advanced therapeutics practice, there wasn't a lot of interest from large entities in buying into the space. However, there was clear proof of concept in small studies, both in the CAR T side as well as in genetic disorders. And so it was clear that, on the horizon, this was all going to come together. And what's really been driving it in the non-malignant side of things is really advanced understanding of biological pathways, very tractable monogenetic diseases where, there's a clear therapeutic strategy of gene augmentation or of loss of function mutations or, knockdown of gain of function mutations or dominant negative disorders, but also because the viral vectors, at least AAV and lentivirus, and potentially, self-inactivating gamma retroviral vectors, appear to be a platform for, for a clinically and regulatory, sort of endorsed platform for new therapeutic approaches. So, as Steve mentioned, now we open up potentially, hundreds of different disorders, from the very rare to potentially large indications that we have some of those on the horizon that we can maybe touch on towards the end of this. What is the broad, how do we reach the broad, patient segments? So we've addressed biological risk, technical risk, translational risk. With the demonstration of, Luxturna getting approved on a very rapid timeline. We have AVX-101 getting RMAT designation and potentially, accelerated pathway to approval. That, now, it seems like the risk is really backloaded. It's different than what it was with conventional therapeutics. Risk is all around the commercial model. How do you scale up manufacturing? Right now, there's, significant limitations to both, internal development capacity, but also in the contract manufacturing side of things. Logistics, distribution, reimbursement, risk-sharing schemes, all this, right now is incredible uncertainty into the commercial model and how that's going to affect the life cycle of products and, potential, longevity of franchises about how they deal with each of these risks. And one more thing I want to touch on before moving back. Also, we are focusing on monogenetic diseases. You have a very different sort of forecast approach, given that many of them are focused on ultra-orphan indications, where you may have a very receptive and motivated patient population that, upon approval, you have rapid uptake into the market. If you take the case of Luxturna, for example, for RPE65 inherited retinal dystrophies, that product's likely to peak in 4 to 5 years into the prevalent patient population, and then we expect a rapid decline as you're waiting for new patients to move into the treatment setting, new incident patients. So we have a very different sort of portfolio strategy when we think about moving forward, regardless of the incredible breakthroughs that we've seen scientifically and clinically.
Yeah, absolutely. And I think that then is a nice transition. And maybe the way to touch upon each of these different topics that I have laid out here and questions is rather than kind of go through each of them in a vacuum or in isolation, why don't we talk about some of the different cases? And Steve, I'd like to start with SMA. I think that's a fascinating one. Obviously, very high profile. The data from Avexis is striking. It triggered a very large, unprecedented historical deal with Novartis, acquiring them. Already with a product in the market, albeit one with a different value proposition and a different therapeutic modality, that of Spinraza. Can you tell us, Steve, maybe just start high level and then get into some of the details about why SMA is a success story from the technical side in terms of alignment of the therapy with the disease itself?
Yeah, no, there's so many things that I can touch on using SMA as an example, but the overview is this is really one of those pivotal studies, pivotal proof of concept studies in the field of CNS gene transfer that I think really woke up the field and showed that gene therapy really was, basically it's for real in the CNS space. So, but the details around this are, one, that I think that the preclinical studies actually did advise and predict this clinical benefit pretty well. I think that's an important point. And, really, the approach here was to do an intravenous injection of AAV9. And that's something where, with that approach, it was really amenable to dosing infants in this disease, and there's a favorable risk versus benefit, kind of analysis on this, but where, typically going into an intravenous approach to target the brain, the manufacturing demands are very, very high because you've got to, you really have to dose this up, pretty substantially to get enough vector into the brain. But since in this case the infants, are obviously very small, then that manufacturing demand was manageable. Taking SMA as an example, you see, that this was a treatment for type 1 SMA in infants. Now this is moving on to larger SMA patients, the type 2 and type 3 patients. But, an observation here is that those approaches are using an intrathecal route of administration. And that's something that's, basically the same vector. And, you can kind of make some inferences there where an intrathecal injection would use a lot less virus. It's been shown to target the spinal cord still very well. And so you can even see just within SMA how the treatment is evolving, where the IV approach works in infants, and it should work in larger patients, but maybe due to manufacturing logistics, as much as anything else, it's moving to an intrathecal route. Yeah, and I think, taking SMA as an example, obviously there's some biology around this disease that is somewhat unique. The targets are fairly clearly spinal cord motor neurons. And so, I think there's a lot of excitement translating this conceptually to other diseases, but there are also differences with other diseases, where you might need to target more broadly other cell types than just spinal cord motor neurons.
Right. So does this, initial success in type 1, and then there's some expansion potential into type 2, type 3, with some incremental changes, not to say that going from an IV to intrathecal is not trivial, but, beyond that, and Mike spoke to kind of the sustainability of curative approaches in small patient populations. Do you see, Steve, and then I'll let Mike speak to this as well, going from SMA to, a related disorder, if such a thing exists, as a way to leverage synergies within a core competency that the company has? Or are we talking about, each disorder, once you move away from SMA, being kind of its own unique situation with its own unique set of technical considerations for optimal positioning?
Well, look, I think one of the key things that you need to go back to is that the preclinical studies really did guide and predict the clinical studies, and the success that was observed in preclinical models did translate to humans. And that's a key basic concept that I think, just tells you a bit about how the vector is working. So even though, you might look at other diseases and the biology is a little bit different from SMA, from the technical aspect of just being able to deliver a gene in a mouse and being able to deliver a gene in a human, that proof of concept is now established. So, if you have good proof of concept in animal models, I think that we have I think it has good predictive value of human efficacy.
Yeah, absolutely. And is that, something that you see as generalizable beyond SMA? It sounds like the answer is yes. Yeah, I think so. The SMA obviously is a huge success, and the efficacy that they saw was truly transformative. I think that we are, watching other clinical trials, other CNS-directed clinical trials to, really sort of verify that is translating across other diseases.
but I think right now it's looking very, it would be very encouraging. And if you talk about other diseases, one aspect is lysosomal storage diseases, where you get cross-correction, that, these are diseases that have been treated with enzyme replacement therapy in some cases. I think that in the gene therapy field, these are viewed as sort of the low-hanging fruit. In a lot of aspects, those might be even easier to treat than spinal muscular atrophy, because, it's not limited to being a cell autonomous effect. You do get the cross correction. So that's, if we take the gene transfer itself as a given, then the biology, you can pick apart and put diseases into certain bins of low-hanging fruit, and then maybe another bin of encouraging, then another bin of possible but kind of needs to be verified.
Right.
And then certainly other bins of, the biology doesn't really line up, and it may be too difficult, that, AAV9 may not be efficient enough.
Mm-hmm. And Steve, when you talk about lysosomal storage disorders as being potential low-hanging fruit, are you considering the validated replacement markets right now, Fabry, Gaucher, Pompe, MPSs, and others, or are you considering other, IEMs, lysosomal storage disorders, that have predominantly CNS effects? Or I'm just trying to understand, when you say low-hanging fruit, are you considering displacing, relatively mature therapeutic markets, or are you talking about opening up new diseases?
I think both. I think it has a potential to display existing markets where enzyme replacement therapy has been, validated. It's a valid approach, but then it's opening the door to those lysosomal storage diseases, inborn errors of metabolism, that are really CNS focused. That now these are opening up a new door to being able to treat those.
Mm-hmm. Okay. Now, going back to the SMA, example that we started with, and, we could even consider CAR T therapy for relapsed refractory pediatric ALL, and others, where, clearly the therapeutic potential, is really demonstrated, cures, or yet to be determined durable cures, in some cases. So, is that setting the precedent that we're going to consider each of these? You mentioned some have more complicated biology. Should this be what we expect for each of these, or would there be, other sort of, therapeutic benefits that also, have, value to patients?
Yeah, I think this is another lesson learned from SMA, or something that we should be thinking about. When we think about the SMA treatment, we think about those most dramatic results in terms of benefit, that, some of the visuals that many of us have seen either in the publication or or various presentations of this. And those really truly transformative benefits in the best patients in that trial. It's easy to have that set expectations and set a bar that may be a little bit unrealistic. And we have to keep in mind that for the majority of these CNS diseases, there's basically nothing out there at all. And so our bar in terms of calling something as a success, that bar should really be set as providing a meaningful benefit to the patient, and not, even if that type of benefit falls short of a quote-unquote cure.
Mm-hmm. Right, that certainly was the case, at least with RT65 with Luxturna, that, we're not, having a hard, mortality benefit. However, the benefits of daily, function and being able to lead a normal life, and indirect benefits and everything were highly rated when they went up for ICER review, after the public comment with stakeholders there.
Yeah. So from the business model perspective, Mike, It sounds like there's a spectrum of value propositions that one could regard as opportunities, whether it's, in terms of internal R&D decision making or external access to external innovation and thinking about which programs to onboard. How do you see companies thinking about this? Do they kind of see the bar set so high that if you can't potentially cure the patient, given that a lot of these patient populations are small, that you don't want any ambiguity in your regulatory package, in your market access package, and ultimately the dossier that physicians are looking at?
Yeah, so I mean, I think it all depends on which disorder we're talking about, right? And you and I also spend a lot of time in oncology, and there clearly there are different benchmarks for adoptive cellular therapies. But for monogenic diseases, I think there's all sorts of benefits, and I think since Scott Gottlieb has been very active with RMAT designation, working with biotechs early innovators, helping support some harmonization across CMCs and some guidance on manufacturing scale up and all this sort of stuff. I think there's less sort of uncertainty on the dossier and value that you're bringing in, as long as you're bringing in a clear therapeutic benefit that's aligned with the patient unmet need, whether that is restoring sight or maintaining independence, or in a case of SMA and these terrible life-threatening pediatric disorders, that you're improving mortality. So I think it really depends on the patient population, but clearly the FDA has the stance now that if you're addressing a significant unmet need, that you're going to be reviewed based on the therapeutic index of safety and efficacy of the program, very similar to conventional therapeutics.
So Steve, maybe moving on, I'm sure we'll come back to it, but moving on from SMA, I suspect the mantra should be, if you've seen one monogenic disorder, you've seen one monogenic disorder, right? And so even within that limited set of disorders, of which there are in fact many, can you talk to us and maybe with reference to a different disease, whether it's Rett syndrome or DMD or something else, where again we're talking about a single gene product that needs to be somehow fixed, whether it's added in or knocked down or edited, and maybe contrast that a little bit with SMA and what the considerations are from a technical standpoint in order to think about how best to position oneself from a therapeutic perspective.
Yeah, so look, I can, there's a few points I can make, and maybe the first would be to compare SMA and Rett syndrome. In the case of SMA, I think there was an abundance of preclinical data saying that overexpression of SMN1 appeared to be safe and well tolerated. So there wasn't this risk of overexpression where this needed to be titrated very carefully. I think Rett syndrome is, kind of the opposite example of that, where, it's known, just in the disease biology that you really need to avoid overexpression. This has been shown in, patients that have duplication syndromes. You have a number of, basic science studies that have looked at overexpression, and then also gene therapy studies, preclinical that have been published. So, that would be a situation where, with Rett, even though there's biology that this is this could be a reversible disease, and in some ways it's a very attractive target because it looks like it is reversible and it's not necessarily degenerative. So there's a good conceptual basis of being able to rescue the disease, but it represents its own challenges, really around regulating the expression of the gene.
Yeah, and I think it's an interesting segue there too, that in that case and other disorders, right? We used to call our advanced therapeutics practice complex therapeutics, right? So, can you sort of maybe elaborate a little bit on what sort of going? So we have a viral vector, we have a therapeutic gene, we potentially have a promoter technology that is regulatable to make it to get the therapeutic index proper for the disease. Maybe you could elaborate a little bit more on all the sort of features that might come together to enable a therapeutic, such as something for Rett disorder.
Yeah, so I think in the field, maybe one of the objectives across the field has been, for a number of diseases is, can we design a vector that we can put in and then modulate the expression with some exogenous drug? It's something where you're basically just putting in the initial expression cassette and then regulating it over time exogenously. So, that's been an objective, but I think even though there's been research papers published on this, I think that there's nothing that's really come to fruition, and it remains a difficult challenge for the field. So there are a number of different kind of technical strategies to regulate expression, but I think right now, in terms of what is feasible with existing, kind of validated technology, is using, say, promoter controls to modulate high expression or low expression, neuron-specific expression, or ubiquitous expression, muscle expression, using promoters, but not necessarily to really finely tune expression levels as you would need for a disease like Rett.
Is that something, if you don't mind me jumping in, is that something that would need to be, if there's a rheostat that we're talking about, is that on a per patient basis, or even within a patient on a longitudinal basis, where there are going to be times where you want more or less of the therapeutic to be actually on board?
Yeah, I think all of those cases could be present depending on the disease. So this is where, when you talk about initial diseases and where this is a platform technology, I think that the easiest diseases are ones where overexpression of the gene doesn't represent any problem. Those are clearly the easiest, safest targets. And then you have to break it down into, again, do you get kind of cross-correction, where if you target 5% of cells, the protein is secreted and treats 100% of the cells. So you can sort of put together an algorithm of easiest to hardest, and then start to put diseases into different bins of, where you really can look at a platform approach of this group of diseases could all be treated the same way using the same technology with a high prediction of it working. Maybe I hope that makes sense.
Yes, certainly. Yeah.
And then you were talking about the examples of other diseases. One, we have Duchenne's muscular dystrophy on there, and this is a different type of problem that's more of a logistical consideration, where I think that there's been preclinical data floating around for a number of years saying that you could treat DMD effectively using AAV vectors, and the biggest obstacle, in my view, the biggest obstacle to making that a clinical program has been around manufacturing. And, because the doses that are needed IV are in the range per kilogram of what was used in SMA, but now your patients are much, much larger. So for every DMD patient you could treat, you might be able to treat 20 SMA infants or something like that.
Can you also speak a little bit more to that with some of the recent safety issues that have been, spoken about by Jim Wilson, that we've, seen in some of the solids DMD trial with, microdystrophin, about high titer AAV and, what sort of consequences might this have for diseases like that involve, structural proteins that, you need to basically deliver across the whole body. How significant are those issues, and, what is being done to address them?
Well, I mean, I think that right now the data is still fairly scant, but there's sort of some general assumptions that are being made around, what's a maximum IV dose per kilogram of AAV. And, I think right now you're looking at the SMA trial, the DMD trial, that are really maybe skirting that soft upper limit. And that's, really, that's a safety limit, but it's also maybe a manufacturing limit as well. So they kind of go hand in hand a little bit right now. So I'm maybe that's enough. Maybe I can stop right there.
Yeah, that's fine. Also, maybe you could talk a little bit. We haven't spoken much about other vectors outside of AAV, and maybe we could talk about in the context of some of the limitations that you see with AAV approaches for CNS disorders, and what other where are ex vivo modified HSCs with lentivirus and retrovirus, and where are nanoparticles and non-viral gene therapy approaches, and where do you see them fitting in, and what are the pros and cons compared to AAV?
Yeah, so I think AAV has emerged as maybe a dominant vector for CNS gene transfer, but one of the limitations of AAV in general is that if you're in a dividing cell population, you'll lose expression over time. So this is why hematopoietic stem cells, for example, would be a really poor target for AAV, and that's where you have other retroviral or lentiviral vectors. Are very good in those kind of ex vivo applications, and in that case, there are some of these inborn errors of metabolism where you get cross correction, where these diseases have been treated by bone marrow transplant to some effect, and in that instance, you can use an ex vivo gene transfer approach to basically overexpress the gene in those bone marrow cells. And then there's a good proof of concept that should work in the clinic, because bone marrow transplant already has a certain amount of efficacy, and if those macrophages and microglia in the brain are expressing more of the enzyme, then you should boost the efficacy. And then maybe the final thing when we talk about nanoparticles is there are cases where you might want transient expression of the gene. And that's and that's not really what you would get with AAV or lentiviral vectors. So if you want transient expression, and we can think for an example of maybe you have something that maybe an application is stroke, where you want transient expression of something to create a healing environment and then go away, or gene editing technology, where you're delivering the editase, and there might be adverse consequences of expressing that editase long term. So these are applications where typically I think nanoparticles had fallen by the wayside a little bit, because it was hard to get sustained gene expression. But now you have these applications that are coming up where actually you just want transient expression, and now nanoparticles fit that bill very well.
Yeah. So if I can maybe pull back a little bit, Mike, when When a company is thinking about portfolio strategy and what their core competency should be, and the answer might be different for a small biotech that is, a large part of its business model is being able to provide innovation for large pharma or spec pharma versus pharma companies that have extant footprints in different therapy areas. Can you speak broadly to the implications of choosing a particular competency, whether it's from a technology platform side or a therapy area focused side, where you're really trying to bring in different modalities that can collectively address the therapy area?
Mm-hmm. Yeah. Yeah, so one of the biggest questions we get is from specialty pharmaceutical companies or franchises that have a particular therapeutic approach. They have a long, history and relationship with treaters and patient populations, and they know how to navigate, the commercialization and the reimbursement space, at least for replacement therapies and small molecules and others, which also can be, considerably expensive. I mean, for enzyme replacement therapy and clotting factor therapy, we're talking about, hundreds of thousands of dollars per patient per year. And so they're looking at one-time curative treatments of how does it fit into their portfolio mix. One, what is the threat to their existing franchises? You had up there hemophilia, and some of the inborn errors metabolism franchises earlier, that, there's a clear, threat of displacement of one-time curative treatments, if we realize that goal for many of these established markets. And hemophilia will probably be the first, example where we see that playing out. But also, if you're a biotech that's leveraging a portfolio of, a platform, and you have a lead asset for one particular indication that leads you to a certain therapeutic area like ophthalmology, is, should you focus down on ophthalmology and bring in take on some more, polygenic disorders that might have broader patient populations, but also think about, acquiring or investing in small molecule and biologic therapies, as part of the portfolio mix, because each of them have a different sustainability of their life cycle. And when we think about, sustainability that shareholders and investors want for the future, we have breakthrough therapies and, considerable benefit, but also we need to figure out how we provide more solutions to patients. And in some cases, it goes into diagnostics and devices and other services, because, most of these historical franchises that focus on ultra-rare disorders have a very high touch, and relationship-based model. So it's not just about the therapeutic, it's about many other things.
Right. And so Steve, if I can ask you to put on a commercial hat, but again, with a technical question, what are the competencies that you see as generalizable, whether it's manufacturing, which you said earlier, scalability is a consideration. Is it the modality itself, whether it's the type of payload or the type of vehicle, which will presumably be evolving very rapidly, and AAV9, as I know we've discussed in the past, may not ultimately be the optimal AAV vector as directed evolution and rational design continue to generate improved technologies in terms of tropism or robustness of the payload that's being delivered. What are some things that you think, as if you're thinking about putting yourself in the perspective of a company and trying to build out a platform, what is a platform in this space? What does it mean?
Yeah, well, look, right now when we talk about AAV9, I think you can take it as a given that there will be better vectors than AAV9 if they don't already exist. And so, but what we're looking at right now when you have a vector like AAV9 is basically it's been validated across multiple models, multiple labs, multiple companies as kind of a gold standard. So it's something that there's across the field. There's a lot known about it. It's a fairly known quantity. So in diseases where it's good enough, then it's a good choice because you can have, from a manufacturing perspective, each product is its own product. But if you have 10 different products that are all AAV9, then the CMC on all of those should be very, very similar. And there may be, I think from a regulatory perspective, it remains to be seen if there really is an economy of scale from a regulatory perspective of different products that all use the same process. But I think that there's room for that. Certainly on the business development standpoint and everything, if you know how to make one AAV9 vector, then you should be able to make 10 others. Now, once we move into better vector technology, which will happen, then each one of those, then you're moving out of that platform, and so there's going to be some investment, I think, reestablishing that platform, and then it may be, maybe you can use that for 50 other diseases. So, I think that's sort of how I see it evolving, and my opinion on this, when you look across the scope of the field and new vector technology, is I think one of the hardest things is that a lot of this is becoming kind of locked up and confidential and proprietary. And you had AAV9, which was used very broadly in an academic setting where you had lots of labs generating lots of data, so you had lots of independent verification of what it can and can't do. A lot of the new vector technology is going to be probably more locked up, where you're just having information on what it can do coming from, one source or a couple of sources. And so it's going to be really hard across the field to pick out, truly what are those better vectors, which ones are the best. I think everybody's going to have their pet vector. And I guess if they work, great, but it's going to be a little bit harder to have that true sort of quote unquote platform.
Yeah, and maybe Mike, I'll put it to you as well. The idea, at least in my mind, the idea of IP is being completely upended as we have continuous innovation, and we're seeing this on the oncology side with CAR T therapy, and you have Carl June, who's a big figure on the oncology side, saying this is going to be like the iPhone, where you get a new improved version every 6 months or a year. What does that then mean in terms of from a business model perspective, in terms of carving out exclusivity and creating barriers to entry, and ensuring that your investment in a particular space is going to generate the proper return?
Yeah, it's a good point, and I think that's why biotechs are spending so much money to try to acquire experienced manufacturing people and scientists and technical people right now, because I believe that the patents on AAV is up in the near future, and I'm sure lentivirus isn't too far behind that. But what is really driving the value is, again, what we were talking about earlier, all the sort of features, novel features that you put in to achieve the therapeutic use, along with the special designations for these rare patient populations, right? But in a sort of evergreening sort of model, if we track the parallels of what's going on in the CAR T space, is that there's the first to market, products right now, Yescarta and Kymriah are not going to be the drivers of the market, maybe even in the next couple of years, because there's so much know-how and additional sort of incremental advancements that are being, engineered into those therapies. And the same will play out in monogenic diseases. However, it's going to, have to be tried to figure out how this all gets streamlined from a regulatory standpoint, because, the FDA is going to be dealing with a lot of different, requests and whatever. So again, I think this is a little bit of risk that there is such a bottleneck of, how do we streamline the innovation, right, and make it accessible to patients, right?
Yeah, and this is Steve. I'm just going to jump in with one other point, because, I think that if you're making that investment in new vector technology and it's less of a known quantity, if you're moving into an area of a rare disease that is still somewhat more common, like SMA or DMD, then maybe the business model may still be attractive for that. But if you're really in what we'll term as an ultra rare disease, where you're talking about maybe less than 100 patients, I think that obviously the business model around that is a lot more complicated. But if you are using a tried and true platform approach, then those are areas where you might be able to take advantage of somewhat of an economy of scale or an established track record, where maybe the cost of moving that drug to market can be less using sort of a known approach or a known quantity. So maybe sort of a divergent thing where you have new vector technology going towards things that are maybe larger patient numbers, more attractive from a business perspective, and then on the other end, it's almost more the bargain approach, where you're using established vector technology and then just trying to streamline your commercialization process to make it where less and less patients are needed to make that model work.
And Joel, I don't know if you're heading this way. I guess the next question I have, around that, if you're talking about the novel vectors opening up larger patient populations, maybe. So, where are we in sort of, advancing gene therapies towards, proteinopathies, aggregation disorders, broader things like ALS and Parkinson's disease? I mean, because this whole conversation so far has been all about ultra-rare monogenetic diseases, but where are we in, establishing larger markets?
And I'll just maybe build on that, because that's exactly where I was going, Mike. Maybe Steve, since it is a question for you, we're no longer in many of these cases, aside from maybe some of the genetically defined subsets of larger disorders like Parkinson's or ALS. Now we're talking about the disease pathology itself, whether it's dopamine secretion in the synucleinopathies, or plaque formation, which still remains an open question as whether and when removal and which molecules need to be removed is going to be disease modifying. We're talking about perhaps the same umbrella of therapeutic modalities, but I suspect we're talking about a completely different animal from the technical perspective. Do you want to maybe provide some high-level thoughts about that, Steve, and then I can continue to ask some drill down questions?
Yeah. Sorry. So I think on the whole, that people are going after the monogenic diseases because those are situations where, from a disease standpoint, we know exactly what needs to be fixed. We know exactly what gene to deliver. That aspect of it is very straightforward. So, the unanswered questions are really the gene delivery technology, and maybe at what point we're moving in the disease course, like how early you have to treat. So, you can view this as really approaching it scientifically where, for the monogenic diseases, we're removing one variable because we know that we're delivering the right gene. If you move into more complex diseases, polygenic or things that are maybe not genetic based treatments, then, if it fails, you don't know, was it a problem with the general approach and what gene or piece of DNA was being delivered, or was it a problem with the actual gene transfer technology? So the way that I think I see this evolving is you have the initial proof of concept with the monogenic diseases to really settle the technology and make it more of an established, just general medical approach, and then that opens the door to treating more complex diseases. I think in a better way.
Right.
And in terms of the approach, we're talking about gene replacement approaches, but there's plenty of approaches that have been, that are being pursued, say for Huntington's disease, where it's a knockdown approach, and that can be done with ASOs, but it's also pretty straightforward to like express an siRNA or shRNA off of an AAV vector.
Right. Yeah, and maybe we could bring the discussion back to what we were originally talking about in terms of the therapeutic modality itself. So if we're talking about AAV9, you want to make sure that you understand what the target tissue is, whether it's specific neuronal subpopulations within the CNS, and optimizing getting the drug there, which speaks to route of administration and also inherent properties of the vector itself. And of course, the more invasive you are with your administration approach, I presume that the higher the bar is in terms of what you need to demonstrate from a clinical profile perspective. But at the same time, there are certainly a lot of people thinking about it on both sides of the blood-brain barrier, for example. So do you want to just kind of speak to maybe a little bit more technically what are some issues that you think should be top of mind?
So top of mind, and sorry, just in terms of gene delivery or general approaches or
gene delivery. So let's talk about Parkinson's. So getting the therapy presumably into a particular part of the brain, the putamen and substantia nigra, and the right cells that are secreting dopamine or perhaps interfering with the metabolism. What are so I guess what are some thoughts around that from the technical side?
All right. Well, maybe Parkinson's is actually a really, really good example because and this is going on this theme. You go to the approach that's being taken for Parkinson's to inject the putamen. There was an approach to deliver the AADC gene for Parkinson's, and I don't know the exact results of that trial, but it was but you basically had now a disease where patients are born without that gene, and you had this AADC trial that was published out of Taiwan that is showing really transformative benefits from just a single a bilateral putamen injection. So this is something where maybe it was more muddy in the context of Parkinson's disease, but when it moved to a monogenic disease, then the results were very, very compelling, and that proof of concept was established. But now, coming back to now, where are we with Parkinson's disease? I think a lot of what that's boiling down to is, one, is the approach, is the gene that we're delivering appropriate? And I think that's, there's a lot of caveats with that with the preclinical models. So this is really, I think, being, the clinical trials are the true experiments here. And then, of course, the outcome measures in Parkinson's and the placebo effects can complicate things quite a lot. And also the Parkinson's is a degenerative disease. So, I think you have to establish, do you have to treat very, very early in the disease course, or is there any prospect of benefit in a later stage Parkinson's patient? But on the whole, I think Parkinson's is an area where there's, certainly a lot of excitement. There's a lot of different approaches. I think that, and that's also something where it's amenable to AAV and it's amenable to lentivirus, because you're just injecting and administering it somewhat locally. So the manufacturing burden isn't very high. I think the injection technology is becoming more and more standardized. And so it just boils down to, is the gene you're delivering correct? And are you intervening at an appropriate time point in the disease course?
Right. Yeah, and it speaks to, I think, the timing of intervention with a large disorder like this then gets into questions that the oncology field has been facing for some time, which is, the time to position oneself is generally later in the pretreated patient populations, perhaps work their way up through the treatment algorithm. There were always open questions about whether a salvage setting patient would be amenable to anything other than just trying to improve survival by weeks or a few months at best. Maybe immunotherapy is upending that paradigm. But it also, from the business model perspective, Mike, if one does need to go into the prodromal or the earlier stages of disease, then you have to think about what the value proposition is, not only to patients, but in terms of the burden that itself could put onto the healthcare system, weighed, of course, against the burden that it's already under.
Mm. Yeah, certainly a dilemma. I think we have a long time to get there yet, but it depends. There may be a situation that one of these programs moves forward earlier. I would put it in the context maybe of what I've seen going on in heart failure, what I've seen going on in atherosclerosis. That there are programs that are going after very large disorders, and they're going to be, as I said earlier, they're going to be evaluated based on their therapeutic, value, really. And what we learned from, the PCSK9 antibodies and dyslipidemia, incredible breakthrough in science that came from, population genetics, and very quickly translated into, incredible LDL lowering therapies and clinical development, and showed outcomes benefits. But at the end of the day, because they're in a broad patient population, they're getting valued based on their number needed to treat to offset, a particular event. I know that Parkinson's disease, is a little bit different than that situation where, you're not necessarily having an event and, having, heart failure related to it and, and morbidity and mortality, but in the same way. But in that case, it comes down to basically a sort of a crude calculation of how many patients do you need to treat, what's the cost of therapy, and what is the cost of avoiding that event.
Yeah. So we have exactly 1 minute left. We do have some great questions that have been coming in. Unfortunately, due to time constraints. So Mike wants to ask one question.
We do have, I think Joel and I can field most of the questions, but we do have one that probably only Steve can answer. And it speaks to the relevance of preclinical models and for CNS disease models. And I assume we're focused on monogenic diseases, but I guess we could also be talking about these broader disorders as well. Is how do you choose and when is the time to use a human gene, I guess, in your gene therapy vector versus the species-specific gene relevant to the model?
Yeah, all right. So I think maybe some of that is risk management. The perfect study design would be to test the human and the mouse gene in a mouse at the same time, in parallel, and then you really generate that data. Usually that's not done. I think you just really have to look at what's the conservation of function across species. If it's a gene that's very highly conserved, then there's probably a reasonable confidence that it's okay to use the human gene in the preclinical models. But of course, one thing is to look at, especially if you're moving into non-human primates, just making sure that there aren't any immune responses if some of the epitopes are different, because I think the immune responses, if anything, could cloud your efficacy and safety results if the gene is viewed as foreign. No, I think generally speaking, the burden is on you from a regulatory perspective to say, what the equivalence is of the human gene and, whatever animal model species you're working in. If you can come back and say that they're equivalent and provide a strong rationale for that, then I think it's appropriate just to use the human gene in all of your studies. That's my opinion, but from a risk management perspective, that just has to be, that risk has to be managed, I guess. And the safest approach is to test both, but that seems like a little bit overkill in my opinion.
Okay, well, it warms my heart that we finished on a very technical translational question, and hopefully everyone who's still hanging on enjoyed the discussion. Again, slides and audio will be available on our website shortly. Let me just thank my panelists, Stephen Gray and Mike Rice, and thanks to all of you for hanging in well past the top of the hour. Take care.